Hello everyone, my name is Landry and I am a professor of biology at Université Laval in Quebec City, Canada. Today I would like to tell you about the work of a postdoctoral fellow working with us in the lab, Dr. Diana Asensio. Diana joined my laboratory to study gene duplication. Gene duplication is one of the most important changes that can occur in a genome during evolution. It is known to drive adaptation, to have contributed to innovation and to the complexity of cellular networks, and it makes cells robust to mutations. For the past 50 years, a lot of attention has been dedicated to explain why gene duplicates are maintained on the long term if they are initially redundant. The two models that are usually explored are, first, the gain of function by a paralog and the retention of ancestral functions by the other, a process called neo-functionalization. And the other model of retention is through the partition of ancestral functions by sub-functionalization. These two models explain the long-term maintenance of gene duplicates. However, what we are interested in this project is the short-term impact of the gene duplicates. The reason why we are interested in this is because the long-term retention highly depends on the immediate effect of a gene duplication. For instance, the duplication of a gene, if the duplication of a gene is deleterious, it would be unlikely to be maintained long enough for gains of function to occur, even if there were advantageous mutations available in the future. The reason why gene duplication can be deleterious is that there is a relationship between fitness and expression level. For most genes, we do not know what this relationship is, but we know that optimal expression is usually around the wild type. We also know that cutting by half expression level leads to decreased fitness for many genes. It has also been shown that overexpressing a protein by several-fold can be deleterious to various degrees. What we do not know, however, is what happens when the duplication occurs. Do we see that the duplication is beneficial? Is it neutral? Or is it deleterious? The main reasons why gene duplication could be deleterious are the following, although there are many others that one could think of. The first one is that producing extra transcript and protein could in itself be an energetic burden for a cell. It's also possible that increasing the amount of a protein could lead to spurious interactions of that protein with others, reducing their function, therefore leading to toxicity. Finally, increasing the expression of a protein could alter the dosage balance among protein within protein complexes, thereby leading to the production of non-functional complex assemblies. In this project, we are particularly interested in this potential mechanism. What is known about the sensitivity of protein complexes to changes in gene dosage goes like this. First, genes whose reduction in expression is deleterious are enriched in protein complexes. So protein complexes appear particularly sensitive to the reduction of gene expression. However, genes that reduce fitness when overexpressed are not overrepresented in protein complexes, suggesting that protein complexes are not particularly sensitive to increased abundance of its subunits. However, there are many limitations to this data and these analyses. First is that most studies are performed in yeast strains that overexpress proteins by 10 to 100-fold, which may produce effects that are different from a simple gene duplication. Secondly, there have been very few attempts to directly measure the effect on protein complexes. We also lack a mechanistic understanding of why an excess subunit would not alter the function of a complex. In this project, our goal was to fill this gap of knowledge. So what Diana did was to design an experiment in which she simulated experimentally the duplication of about 900 genes, one by one. She focused on essential genes because they typically have no recent gene duplicates, and they are enriched in protein complexes. To simulate duplication, she introduced in yeast strains an additional copy of a gene on a single copy plasmid, and this gene is regulated by its own promoter and other regulatory sequences. The strains were marked with M-cherry, and another strain bearing an empty plasmid was marked with cyan fluorescent markers so that she could follow the ratio of the two strains through time. In a competition experiment, by following the ratio of the two strains through time, she could estimate the fitness effects of each of the duplications and measure the selection coefficients associated with these gene duplications and identify beneficial, neutral, and deleterious duplications. What she found is, or what she could measure, is the following distribution of fitness effects. What you see in blue are gene duplications that are beneficial above 1% level, so they have a selection coefficient above 0.01. What you see in red are cases where gene duplication is deleterious. They have fitness effects smaller than minus 0.01. And in yellow are the ones that fall in the middle. The black dots here represent an experiment in which she computed the empty plasmid against itself to have a measure of noise in these experiments, so we can set conservative thresholds and cutoffs to identify which duplications have significant effects. What you can see from this is that there are many more duplications that have a negative effect on fitness, and these have much stronger effects than the beneficial effects. So if we use this conservative cutoff of 1%, what we see is there are twice as many deleterious duplications as there are positive duplications. One of the very deleterious duplication is in the gene, the duplication of ACT1, which codes for actin. And actin is one of the most abundant proteins in the cell, and it is known to be very dosage sensitive. There are some interesting genes among the genes that increase fitness when duplicated. I won't have time to go into the details, but some tend to be, for instance, regulators of metabolism. Now we ask, are protein complexes particularly affected by these duplications? So we look at the distribution of selection coefficients for genes that code for protein that are member of protein complexes and those that code for proteins that are not member of complexes. And what we see is that there's no significant difference between the two. Genes that are within complexes do not tend to be more deleterious than genes that are not coding for a protein that are part of complexes. If we look at protein complexes one by one, we see a pattern like this, where we have on the x-axis several complexes for which we duplicated subunits, and you can see that the median is always around 0. But there are some cases where we do see deleterious effects, for instance, in the proteasome and the RNA polymerase. So for the rest of the project, we decided to focus on these two. The RNA polymerase and the proteasome are very important complexes. The proteasome regulates the active degradation of proteins in the cell, and the RNA polymerase is responsible for synthesizing mRNAs. What we did with these complexes is that we measured pairwise interactions among all of these subunits while duplicating one by one all of the other subunits. So we can measure if there are changes in interactions among proteins when we duplicate genes coding for subunits of the complex. So the experiments look like this. We have yeast cells in which we have two proteins of interest, subunit 1 and subunit 2. and for which we can test protein-protein interaction. And a gene that we focus on here is VPS5 in this example. So we can have a strain like this with an empty plasmid, like we did for the competition experiments, and another strain where we also measure the interaction between S1 and S2, where we duplicate VPS5. So in this case, we're not focusing on the proteasome or the RNA polymerase. We're just testing, showing that the system is working using a small complex called the retromer. So in this particular assay that we call the DHFR-PCA assay, the strength of the interaction is affecting growth of yeast cells, so we can actually measure changes in protein interactions. So you see an example here of an interaction between two proteins in a wild-type background, and the strength of an interaction between two proteins in a background where we duplicated VPS5. You can see that the colony size is bigger, and we can measure those and infer changes in protein interactions. The way we measure changes in protein interactions is called a perturbation score. So this is a matrix that measure the changes in interactions. When the perturbation score is positive, it means that the interaction is increased, and when it is negative, it means that the interaction is decreased. So we tested that to see if the system works, we tested these experiments to see if the system works on the retromer complex, which is a small complex composed of 6 subunits. So you see this, the whole network of the complex here between the 6 subunits as in strain S1, and of the 6 subunits in a strain we called S2. And what you can see here are the results when we duplicate VPS5. And you can see that we do detect changes, increased interactions, for instance, between VPS26 and VPS29, 2 subunits of the complex. And the other thing that you see here are cases where we see decreased interaction, and all of those cases concern cases that involve an interaction between VPS5 and another protein. And the reason for that is that if we duplicate, in this context here, VPS5, it's going to compete with VPS5 that we tagged with the DHFR reporter, and will decrease the strength of the interaction. So this is completely expected given the way we do these experiments. So the experiments work, and now we looked at the proteasome and the RNA polymerase. What I'm showing here on the graph is the cumulative distribution of perturbation score. So you can see it is centered around 0. And we can see some losses of interactions that are very weak changes, and some gains of interactions. But overall, there are very few protein-protein interactions that are affected. So if we look at all the combinations that we tested of duplications and protein interactions, there's only 46 out of the almost 9,000 combinations that show significant differences. And those interactions that are affected usually occur in a background of a protein that is part of the proteasome that is called PRACE7. So now we can look at the correlation between the selection coefficient we measured in the competition experiments and the changes in interaction that we show here on the x-axis as the absolute perturbation score. And we can see a negative correlation between the two that is significant. However, most of the relationship is driven by this particular subunit that affects protein interactions when it is duplicated, PRACE7. And this duplication also affects fitness, so this creates this negative relationship. So apart from PRACE7, whose duplication affects fitness and also affect protein-protein interactions, we see very little relationship between the two. So why would that be? It seems that overall gene duplication appears to have little effects on fitness in complexes, and it doesn't seem to affect protein interactions either. So when we looked at the literature, we saw that authors have reported that at least in yeast aneuploid cells, that when DNA segments are duplicated, mRNA abundance is almost systematically increased, but protein abundance often remain unchanged. You can see that on this plot. So this is a graph showing a yeast strain that has this copy number variation in one of its chromosome. You can see an increase in DNA content for that region of the chromosome. You can see almost always an increase in mRNA abundance, but what you can see is that for many proteins, there's no change in protein abundance. This phenomenon has been called protein attenuation. So it seems that there is a mechanism that exists in the cell that regulates the amount of protein and brings it back almost to normal level, even when a gene is duplicated. So we set out to measure that in the protein complexes that we studied. We focused on the proteasome. The way we can measure protein attenuation is the following. So we imagine that we have a yeast strains in which we have fusions, GFP fusions for all the subunits of the protein complex. And as we did before, we can transform these strains with an empty vector and measure GFP expression. When there is no attenuation, we expect that if we add an additional subunit of that, an additional copy of that gene coding for the subunit 1, we will have an increased expression of that subunit here, but the GFP level would remain the same because the overall expression increases, but half of it will come from the genomic copy. However, when there is attenuation, we would expect a reduction in protein abundance so that it matches the wild type, or at least partially. And then because attenuation would occur for both the GFP copy and the non-tag copy, we would see a reduction in GFP signal. So we tested if there was protein attenuation in the proteasome complex, and we found that most subunits are actually attenuated. What you can see on this plot is GFP expression in wild-type strains where there's no duplication, and in strains on the y-axis where there is a duplication. And if there was no attenuation, we would expect all the points to fall along this diagonal. And what you can see is that there are many cases where expression level in the strain with the duplication is actually lower than in the strain without duplication, strongly supporting the model where there is in which there is attenuation of the subunits when we duplicate the genes coding for these subunits. One interesting case is the subunit that affects fitness and that affects protein-protein interactions, pre7. And pre7 is also attenuated, but it is one of the most highly expressed subunits of the proteasome. So we suspect that this is why, even if it is attenuated, it does impact fitness and protein-protein interactions. So it seems that protein complexes are not sensitive to increase in protein abundance and gene duplication because the excess protein may be degraded instead of causing problems in protein complex assembly. In conclusion, what we found from these experiments is that gene duplication is more often deleterious than beneficial, which means that many genes have little chance to be maintained after duplication. But this doesn't seem to be particularly deleterious when it happens for genes coding for protein that are part of protein complexes. Also, it is quite interesting to see that a mechanism that may be there to control protein complex assembly may at the same time protect from the consequences of gene duplication, such as the change in protein abundance. So again, I would like to thank Diana for her work on this project, our collaborators, particularly Alexander De Luna from Mexico, and all members of my lab and the funding agencies for supporting our work.